what we've talked about so far in Chapter 2, we've discussed chemistry, the periodic table. We discussed how it was organized. We looked at that. We also looked at atoms and how they're denoted or signified on the periodic table. So the atomic number, the element symbol. I said atoms, but I mean elements, and again, those were used interchangeably. So the atomic number, which is equal to the number of protons, and the number of protons always equals the number of electrons. Fantastic. Then we have the atomic mass at the bottom, which is always a decimal, which is equal to the number of protons plus neutrons. Okay? Protons plus neutrons. And what if you have an atom that has a different number of neutrons? How do we refer to it? Isotope. Fantastic. So we discussed that. And we also talked about the charges. Like protons have a positive charge. Electrons have a negative. Fantastic. And neutrons have neutral charge. So we discussed that. We discussed the organization of electrons. How many electrons go in that first energy level within that orbit? Two. And each consecutive? 8. So we discussed that as well. And I can't think of what else did we discuss. What else did we discuss that I'm not covering? It's structural formulas. Okay, yeah, structural formulas and how we're going to start to build those. We kind of touched on those. That was actually the last topic that we covered. And I was kind of introing that, and that's where I stopped. I think I'm missing something, but I'm sure it'll come up later. Where we're picking up, and this is why I wanted to stop and pick up here on part 2. Because one of the 7 characteristics of all living things that we discussed in chapter 1 was that all living things have a metabolism. Do you remember what the definition of metabolism is? It's just a series of chemical reactions. So that's where I'm picking up right here. Metabolism is a series of chemical reactions. Chemistry is a difficult subject to teach, but my job is to help you link chemistry to biology. So one of the 7 characteristics of all living organisms is that they have a metabolism. If we define metabolism as a series of chemical reactions, I can make this make sense to you. Because guess what's occurring inside of you? Metabolism, which are chemical reactions. Okay? Which again is why we study chemistry. So, just generally looking at what I have, the information that I have on this slide, I have a chemical equation. Okay? And I have an arrow which tells you whatever's here is going to turn into that. Always in this setup, anything on the left is a reactant, anything on the right of the arrow is a product. And the arrow just tells you in which direction that reaction is going. So reactants go in, products come out. Reactants go in, products go out. Now, of course, I feel like you should know this by now, but just in case you don't, I did not just randomly put up an equation here. This is a meaningful equation. Who knows what this is the equation for? This is the equation for photosynthesis. I chose this equation because, guess what equation you have to memorize? Photosynthesis. Not in this chapter, but when we get to photosynthesis, you will. So I didn't want to give us a random equation. I wanted to give us one that was applicable to something we would study. Now, do you carry this out? No. You don't. Okay? But if I were to give a student this equation, and I gave them a series of multiple choice answers, if I didn't have this information here, very few students would get it correct. However, if I could identify that as being carbon dioxide, which I did, and that as being water, which I did. This right here, C6H12O6, first time I'm going to say it, but you have to memorize this one too. This is the molecular formula for glucose. Glucose, which is our main carbohydrate for energy. You will have to be able to recognize that when you leave my class. You should be able to look at it and be like, oh, that's glucose. but glucose is a sugar. And then O2 is an oxygen. These 6's that are here are so that this equation is balanced, and that's the chemistry aspect. You will not be responsible for doing that. But you will need to know what are these molecules or compounds. If you could label that as carbon dioxide and water, and a sugar and oxygen, and I asked you what equation this was, one person said photosynthesis, maybe a couple more were thinking it, but a lot of students could have guessed that if they would have had this information, because they know that plants produce what gas? Oxygen. They take in carbon dioxide. So plants take in carbon dioxide and they release oxygen. If it was something applying to a living organism that was not photosynthetic, guess where the oxygen would be? On the other side. You and I, our oxygen's over here, and where's our carbon dioxide? On the other side. So my goal is to teach you how to break things down so you can analyze them and get the correct answer, choose the correct answer. Okay? Of course in biology, because I teach biology, but in all different things as well. So in this chemical reaction, we have our reactants that are on the left side of the arrow, our products on the right side. The arrow indicates the direction of that reaction. And I intentionally used photosynthesis because I do believe that this is the easier one to memorize. So, chemical reactions. Now, chemical bonds. A chemical bond joins atoms together to form molecules or compounds. A chemical bond joins atoms together to form molecules or compounds. A chemical bond joins atoms together to form molecules and compounds. We are going to cover, and again, I've organized these notes to chunk this information, 3 types of chemical bonds: ionic, covalent, and hydrogen. And this is the order that I'm going to teach them in. But I want to discuss the strength of each one. And I'll start with hydrogen being the weakest bond. Hydrogen is the weakest bond that you and I will study. This is very easily broken, and I'll explain that when I get there. Covalent bonds are the strongest. Covalent bonds are the strongest bonds that you and I will study. So where do you think ionic fall into place? They're kind of in the middle. Yeah, they're in the middle. And they vary in their strength depending on what atoms are used. And I'll explain as we get there. So we're going to talk about 3 types of chemical bonds: ionic, covalent, and hydrogen, in that order. But in order of strength, hydrogens are the weakest, ionic are there in the middle, and covalent are the strongest. Okay? Start with ionic. Ionic bonds, by definition, form between atoms that gain or lose electrons. They gain or lose electrons. That's going to be the definition. Gaining or losing electrons. So the example that I used for the rest of this unit, let me not say unit, chapter, is sodium chloride. Does anyone know what sodium chloride is? Yeah, it's just salt. It's just the fancy name for salt. So we're using salt. So sodium chloride is composed of sodium and chlorine. Okay? Sodium has the element symbol of Na. It has the atomic number of 11. If it has an atomic number of 11, what does that tell me? It has 11 protons. So I have that here. It has 11 protons. And as a result of having 11 protons, that also means it has 11 electrons. Fantastic. And you and I know that when I organize those electrons for each of those shells, within those orbits, the first one only gets 2. The second one gets 8. So now I have 10. And I'll have to add another shell to house that last one. This atom is neutrally charged, but it's unbalanced. It's neutrally charged, unbalanced. It's neutral because it has 11 positives and 11 negatives, right? There's no net charge there, so it's neutral. But what is it wanting right here? It needs 7 more. It needs 7 more electrons. Yes. Okay, so it's in need. It's neutral, not balanced. So then I have chlorine. Chlorine has the element symbol of Cl and the atomic number of 17, which tells me it's got 17 protons by definition, and we always know that the number of protons is equal to number of electrons. Fantastic. So I have 17 protons in my nuclei, and my electrons go 2, 8 for 10, and then that gives me 7 on the outside. Again, this atom is neutral, but not balanced. It's neutrally charged because it's got 17 positives and 17 negatives, but it's only got 7 electrons in that outer shell, and it wants 8. It wants 1 more. So this one wants 1, and this one has 1. And this one wants 7, and this one... I feel like we could be matchmakers here. Okay? I think you know where this is going. So, what happens here is this sodium says, well, hey, I have an electron here, and you need an electron because it would seem more, I don't want to say more easy. It would make more sense if I would give you 1 as opposed to you giving me 7, right? It would be easier. So sodium says, here, go ahead and take that electron. So because they have no electrons here now, what happens to that shell? It disappears, right? And now this is balanced because it has 8, bless you. And then we gained 1 over here, so that 7 became 8. And now this is balanced. Are these physically touching each other, these two atoms? No. But they're bound. Sodium said, I gave you something really important. We're going to stay together. I'm not going to touch you, but I'm not going to leave your side either. Okay? Like your crazy stalker boyfriend or girlfriend. Right? I'm right here. I'm not touching you, but I'm not going to leave you. I gave you my electron. You're welcome. And I'll be here to remind you that you took my electron. Okay? That's an ionic bond. They gained or lost an electron. In this case, sodium lost an electron, and chlorine gained an electron. And now this is considered sodium chloride. Okay, so ionic bonds gain or lose electrons. Ionic bonds gain or lose electrons. Now, let's talk about these terms cation and anion. Cation and anion. So, when we did sodium, it has an atomic number of 11 and an atomic mass of 22. We know that because the atomic number is 11, that means that's the number of protons, fantastic, which is also equal to the number of, oh, do you see what I did there? In my life, I mess up a lot. Electrons, yes. And then the atomic mass, so this is atomic number. The atomic mass is equal to the number of protons plus, like you add them together, the number of neutrons. So, we know in sodium that the atomic number is 11, so how many protons do I have in the nucleus? 11. Okay, and this is exactly what we had on that previous slide. We also know that if we have 11 protons, we should have 11 electrons. So I'm going to go ahead and sketch this out again. Okay, so I have 11 electrons. Again, we are neutral, not balanced. Over here we have chlorine. Chlorine has an atomic number of 17, mass of 34. And you know what those numbers are equal to. So I have 17, I didn't mean to write chlorine right here. I have 17 protons in my nucleus and 17 electrons organized in energy shells within their orbits here. So I have 17 electrons. Again, neutral, not balanced. So when they decide to form this bond, and sodium says, you know what, I'll give you one of my electrons. So this electron comes over here. I no longer have this outer shell. And how many electrons does sodium have? 10. It has 10 electrons. Is this neutral anymore?
No.
It's slightly what?
Positive.
I have 11 positives and 10 negatives. So sodium right now has a slightly positive charge. So we write it like that. We write it like that with sodium, and then it has a plus sign. This indicates that it is a cation. A cation is a positively charged ion, which just means that I have more positives here than negatives. Does everyone see that? I have more positives than negatives. A cation has lost an electron. Did sodium lose an electron? Yeah, it gave it to chlorine. And then it uses sodium as the example because I want to keep a uniform example. So a cation is positively charged. Okay? Sodium is a cation. Now, I'm going to teach you how I study people I think they're great. Like if you're a really good athlete at something I'm interested in, like volleyball specifically, I will watch your arm swing over and over again. I just study. I heard a chemistry teacher was really awesome, so I wanted to sit in one of her classes, and I did. And I left her class knowing a few things that I did not know when I walked in a 45-minute time period. First of all, her first lesson was the difference between cations and anions. And she said, do you want to know how I remember that cations have a positive charge? And of course, her students were like, yes. She said, this looks like a cat's butthole. And I was like, what? Like, I had no idea where she was going with it. She said, but it's positive, and it's a cation. To this day, I can hear cation on my positive charge. Like, it's there. Like, I was so shocked when she said it. It's stuck in my memory. Cations have a positive charge because they lost an electron. Over here, this fellow over here gained an electron, is now balanced, but how many electrons are here? 18. So am I neutral? No, I have a slightly negative charge, and that's denoted this way. So you have the element symbol and then a negative, which shows you that you gained an electron. We reference that as being an anion. A negatively charged ion. And it's negatively charged because it gained an electron. So it has more negatives than it does positives. Does that make sense? Is there anyone who's confused right now on the difference between a cation being a positively charged ion because it has lost an electron, and an anion being a negatively charged ion because it has gained an electron? And because they say ions, what type of bonds are formed? Ionic bonds. Is there a physical connection? No, it's just a force field. It's an attractive force that operates. Okay? So here is the diagram that I used on the previous slide. It's just enlarged. Again, starting with sodium and chlorine, and then you can see here that sodium lost an electron. It gave it to chlorine, so now it's balanced, but not neutral. And we can see that it has a net positive charge, which means it has more protons than it does electrons. And chlorine is now balanced because it received an electron and has a net negative charge. Which one of these is a cation? Sodium. Which one's an anion? Chlorine. Okay? They're not physically touching. This is an attractive force. So here's a summary on ionic bonds. There's an attractive force between these. This force extends in all other directions, and it varies in strength. So you're not really sure if it's a weak ionic bond or a strong ionic bond. It just varies depending on what atoms/ions we're using there. So ionic bonds. Questions? All right. Now, covalent. Covalent bonds. We are not gaining or losing electrons with covalent bonds. We are sharing. So there will be a physical connection. We will physically be holding on to one another. Covalent. We are sharing electrons. As I will do through the entire semester, I will often bring in terms ahead of when you technically have to learn them because I know it'll help you to teach them now. So covalent, there are two types of covalent bonds, nonpolar and polar covalent bonds. Nonpolar and polar covalent bonds. Again, covalent bonds are sharing electrons. There are two types, nonpolar and polar. We will start with nonpolar. In nonpolar covalent bonds, electrons are evenly shared. They're evenly shared. In this example here, I have a hydrogen. Hydrogen has one proton, so how many electrons does it have? One. How many does it want in that first? Two. Here we have another hydrogen in the same boat. It has one and wants two. This guy says, I need one. This guy says, I need one. So they share. When you look at this right here, it has two electrons. And when you look at this right here, it has two electrons. But they are sharing them. They are physically connected. This is a covalent bond. Physically connected. This is a covalent bond. And because those electrons are evenly shared, this is linear. Here's a part that I'm adding that's not on this slide. This usually creates a situation where that molecular compound is linear. And if it's linear, it's hydrophobic. If it's linear, it's hydrophobic. What does hydrophobic mean? It's afraid of water. So nonpolar, linear, hydrophobic. Case in point. When there is gas in water, what does it create? If there's a gas leak in a pipe, what do they put the pipe in to find out where the gas leak is? No? They literally put it in water. And how does the water indicate where the leak is? It makes bubbles. Gas makes bubbles. It doesn't dissolve in the water. It creates bubbles so you can actually see it. It's hydrophobic. Let me give you a molecule that you're more, or a compound that you're more familiar with that's hydrophobic. Oils. If you put like vegetable oil in water, does it dissolve? No. It'll kind of mess around like this, and then if you let it sit there, it will separate. It's hydrophobic. Guess what shape those molecules are? Linear. Guess what type of bonds they? Nonpolar covalent bonds. We're linking all of this. So hydrogen gas is an example of a nonpolar covalent bond that's linear and hydrophobic. Oxygen is the same way, so is nitrogen. So if nonpolar is linear and hydrophobic, what do you think polar is? It's hydrophilic, nonlinear, so it's bent, or nonlinear, yes. So in a polar covalent bond, electrons are unevenly distributed. They're unevenly shared. And because it's an uneven sharing, it's not a linear shape. It's usually a bent shape or some other shape. And because of that, it says here that they easily dissolve in water. We call that hydrophilic. So hydrophobic is afraid of water. Hydrophilic loves water. Hydrophobic is afraid of water. Hydrophilic loves water. Using this example here, we already know that hydrogen wants 2. Oxygen, by itself, only has 8 electrons. Its atomic number is 8. So it has 2 in that first level that we don't have to show, because you can clearly see we're into the second level, and we would only be at the second level if the first level was full. If we didn't fill the first level, we would never jump to the next level. So we are to assume that the first level is full. So we have 2 there, and we have 2, 4, we have 6 here. How many do we want? We want 2 more. We need 8. So here I have a hydrogen that has 1, and I have another hydrogen that has 1. We're not going to gain or lose here because we're not ionic. We are going to share, which makes it covalent. And it's not an even sharing. We're just sharing these 2. Not every single one is going to share. So it's an uneven sharing. So look at this shape of this. It's not linear. It's bent. So it goes from being hydrophobic to hydrophilic. Polar, hydrophilic, nonpolar, hydrophobic. Examples of polar covalent bonds. First one is water. Do you believe that water is hydrophilic or hydrophobic? I would hope that it's hydrophilic because water is, it's water. Yeah, it's water. Water's water. That it is. Okay? So it's an uneven distribution. It easily dissolves in water. It easily dissolves in water. Polar covalent bonds. We're sharing electrons, not gaining or losing. In your notes, there's a hyperlinked video. Of course, these are mine reformatted, but yours also has a video that discusses covalent bonds. I put this up here because I need you and I to be able to interpret what this is telling us. Okay? Right here, these are covalent bonds. How many electrons are being shared here if there's one covalent bond? 2 electrons. For every covalent bond, you're sharing 2 electrons. One from one atom and one from the other. So we're sharing 2. Okay, right here we have what we call single bonds. So we're sharing 2 here, we're sharing 2 here, sharing 2. Okay, and that's denoted by a single line. What's happening right here? We're sharing 4. So we consider this a double covalent bond. And when you build this, you will have to have 2 bonds there. You will have to have 2 bonds there. How many are being shared here? 2. How many are being shared here? 2. But here there are 4. So this is one of those things where I expose it to you now, and then it becomes easier when you're applying it. What's happening here? We're sharing 6 we're sharing 6. Each bond shares 2. So if I have 3 bonds, I'm sharing 6 electrons between the 2 atoms. Okay? Which of these do you think is the strongest? Triple. I consider like single covalent bond like you're holding hands, like one hand. Double covalent bond would be like holding 2 hands. So we're tighter together. Yes, I get it. It's a stronger bond. Triple would be like 2 hands and your foot, right? So like you have control over me because now I'm just 1 foot hopping around. That's it. So this is a much stronger bond. What you need to be able to do is whenever you see it written like this, the structural formula, you're able to build it. So you know that when you look at there should be 3 bonds right here. And you know what that means. If there's 3 bonds, then they're sharing 6 electrons. Like you can interpret that. How many am I sharing here? You see where I'm going with that? We could just play a game. Okay, yeah. Good job, good job, good job. Okay, the third and final type of bond. Not final topic, but final type of bond, hydrogen bond. And I told you the hydrogen bonds are the weakest of the bonds. Hydrogen bonds are the weakest of the bonds. These are very easy to break. When we look at an organization or a chart or a model or a picture or a diagram, hydrogen bonds are usually depicted by dots. So covalent bonds are by these lines. Hydrogen bonds indicates that hydrogen bonds are indicated by dots. Okay, so like a dotted line. Hydrogen bonds. We study hydrogen bonds because they're found in biomolecules. Well, it'll be water and biomolecules. Tell me what a biomolecule is. Is it living? If it's a molecule. What's the basic unit of life? A cell. A molecule is smaller than a cell, so is a molecule living? No. But this is a biomolecule. Tell me what that probably means. It's found or within a living thing. You already know this. I just have to help you think it through. Okay? In life, and this is actually what chapter 3 is about, we discussed the 4 biomolecules that are found in living things. Carbohydrates. How many of you have heard that term before? Lipids, proteins, and nucleic acids, which is, what's a nucleic acid? DNA. DNA. We only have 4 biomolecules. And hydrogen bonds, guess where they're found? In biomolecules. Water, which is what I'm going to teach you today, but also in biomolecules. So hydrogen bonds are a weak bond, but they're used in stabilizing large biomolecules or biological molecules. Molecules found in living organisms, like specifically proteins, are a type of biomolecule. Here, what type of bond is this? Single covalent bond. What type of bond is this? Hydrogen bond. So this is a good diagram where you can see that there's different types of bonds within that structure, in this case a protein. Which is why I have it there. So today my goal is to talk about hydrogen bonds within water. And this is something that you already know, but you just didn't know it in this context. So water is H2O, is one water molecule. And when you put a whole lot of water molecules together and you get like water, it's a chaotic organization. There's a bunch of H2Os in here. And they form what we call a water lattice. So a water lattice, let me tell you where you've seen one before, so you can picture this. If you've ever seen a pond and you've looked across it and it looks like glass because it's so still, that surface tension. If you've looked at a swimming pool and seen that, like where it's just really still. A bathtub does the same thing, a pot of water does the same thing. That is the water lattice, the organization of water molecules, and what type of bonds are holding water together? Hydrogen bonds. How easy is it for us to break one of those bonds? Give me an example of, I'm in a lake, I'm looking at it, it looks like glass. What's something that could break that glassy look? Throw a rock. A leaf drops. A bird flies over and poops. You jump in there. You put your finger in there. The wind blows. Those hydrogen bonds begin to break. Now, can they get back together? Yeah. Hydrogen bonds are easily broken. They're easily created. Okay? So we have our water lattice. So here, it's describing the water lattice. It says that in a water lattice, neighboring water molecules form hydrogen bonds temporarily. What does that mean? Yeah, they're not there permanently. Like these two water molecules are not going to stay together forever. It's just temporary. Okay? It mentions, and here's where it helps you to be able to dissect or interpret what these words mean. It says that in the water lattice, it is difficult for nonpolar substances to penetrate. What does that mean? Okay. What type of things are nonpolar?
Oil, gas.
And those are all described as being hydrophobic. So here it says it's difficult for nonpolar substances to penetrate. Does that make sense? If we have water, does it make sense that water and oil are not going to mix? Yes. Does it make sense that water and gas is not going to mix? Yes. It does. Then the third descriptor is that polar or charged substances can easily penetrate. What's polar? Water. We call it hydrophilic. So if you have water, nonpolar things aren't welcome. Polar things are welcome. That's what this slide is saying. But it's saying it in fancy display of scientific language. What's the difference between water and ice? The obvious difference is it's frozen. Yes. Yes, the temperature. Okay, but is it still H2O? Yeah. It's truly just the organization. So here it's what we call the water lattice. It's pretty chaotic. When water freezes, it immediately associates into hexagons. As soon as it hits that temperature, it looks like this. This is what we call the ice lattice. The organization of water molecules in ice. This is what gives a snowflake its shape. And that's why they're all unique. Because of this ice lattice that forms when water freezes or hits a freezing temperature. They're both composed of H2O. That's a fact. It's the temperature that's the difference. When it's liquid, they're very chaotic and moving around. When it's frozen, they're very organized and structured. In this case right here, I have a lot of free space, and it's evenly distributed. Free space is evenly distributed. I'm going to go back a slide. Here's my water. There's free space. Is it evenly distributed? It's chaotic. And the water's constantly going to be moving and fluctuating. It's fluid. Because of this organizational pattern, this free space allows ice to be lighter, less dense. So what does ice do when it's in water? Flows. Ice and water are H2O, just at different temperatures. Because of the different temperatures, the molecules take on a different organizational pattern. If it's a liquid, they're chaotic, still held together by hydrogen bonds. If it's ice, it's frozen, it's organized into hexagons and still held together by hydrogen bonds. Here's water versus ice, the lattice, the organization of those molecules, and you can clearly see that they're still held together by what type of bonds? Hydrogen bonds. Okay? Questions on ice versus water? Okay? This slide you've already written because I taught it before we got here. This slide says, first of all, let's read and interpret this very first bullet, or whatever you want to call it. Raise your hand if you can tell me what that means. Please. At 3. Anybody who's like, I'm not going to call you out. I just want to know, do you understand what this means? Is this a complex sentence? What if you replaced polar with hydrophilic and nonpolar with hydrophobic? Water tends to associate with other hydrophilic molecules, and it excludes hydrophobic ones. You walked into this class not knowing you were going to walk out of genius. No idea. Here, polar, it says, are hydrophobic. I said that in my head wrong. I don't know, in my mouth I said it wrong. In my head I said it right. Polar is hydrophilic, and it means it loves water. So I have it here written, even though I discussed it earlier. Nonpolar is hydrophobic, and it's afraid of water. Okay? That terminology there, where that first descriptor is what you will have to be able to interpret. And I get that we're together and I'm here supporting you, but you have to be able to interpret it when you read it on your own without me prompting you to say, what could we put here instead? Does that make sense? Cohesion versus adhesion. Cohesion is water to water. Adhesion is water to something else. Cohesion is water to water. Adhesion is water to something else. Cohesion is water to water. Adhesion is water to something else. So when I mentioned a moment ago about looking at a lake or a body of water and it appearing to look like glass, that property is maintained by cohesion. Those are water molecules sticking together, and they're cohesive properties, so water to water. In the lab that we did last week, the measurement lab, we used a tool, the graduated cylinder, and we added water to it. And when we put water in this glass graduated cylinder, and it would do the same in a plastic one, and it does the same in your coffee cup, but what did it form? A meniscus was the name of it, and it was discussed in the lab. That meniscus, or that dip, is literally caused because water tries to climb out. And normally I would use your water bottles, for example, but there's no water bottles in our labs out anymore. But when you shake a water bottle and you set it down, does all the water fall back into the bottle? No. There's a few droplets that stick to the plastic. That's adhesion. This meniscus is formed by water sticking to this glass. Curious, if I was going to read the measurement of this water, where would I read it? Bottom of the meniscus, the lowest part of the meniscus, the bottom. That's a test question, so I was going to make sure. Okay, now, how do I remember this? I remember adhesion like an adhesive, so it's sticking to something. So water sticking to something else. And I remember cohesion like co-worker, like they're the same as you. So water to water is cohesion, water to something else is adhesion. Cohesion, adhesion. This surface tension of water that causes it to look like glass, is that cohesion or adhesion? Cohesion. Water to water is cohesion. Now, I'm just curious. Why can this guy stand on this water and not go through it? Because if I stood on the water, I'm going to go right through it. What is he doing that we don't do? He's not floating. He's not hovering. He's actually on the water. He's not sticking. He's evened out. What does she mean by that? So if I step in the water right here, you're going to get 150 right here in one spot. What's this guy doing with his weight? He's distributing it. He's distributing it evenly so that not all of his weight is right here. He's distributing it. When I learned this concept, I thought to myself, so I wonder if I went right through the water.
So, okay, question.
Go ahead.
What's the difference between doing that and floating?
Great question. So floating, you're actually in the water. Like you're not atop the water. This fella is on top of the water right now. And his distribution makes his weight. So he weighs, let's say, 2 grams. And if you put 2 paper clips and dropped it into the water, they would go straight through, right? But because he is distributing his weight in such a way, he's light enough to not break the surface tension of the water.
So I guess it all depends on your mass.
Yes. Oh, for sure it's your mass.
Okay.
There would have to be a large distribution to get me to not go through that. But in my head, I was like, I wonder if I could do it. But it looked like a belly flop. So whatever. You should seriously like my brain works in like, I'll try that. And then I'm like, oh, it didn't work out. Okay. Good question, though. This topic right here, I think is a perfect time to intro this. And then I'm going to bring it up again when we are together on Thursday. So the topic is the lipid bilayer. What can you tell me about lipid? What's a lipid? Fat. Lipid is the fancy word for fat. Inappropriate for you to say you are lipid. Can't say that. That doesn't make sense. Okay? But lipid is a fat. And we will reference fats as lipids, right? Bilayer. What does that mean? 2 layers. Bi means 2 and then a layer. So I have a lipid bilayer. Interpret what that is. 2 layers of fat. This lipid bilayer is also referred to as a phospholipid bilayer, which may also be called a cell membrane, which could also be referred to as a plasma membrane and may even be referenced as a biological membrane. I put them all here because I already know that I'm going to teach you lipid bilayer, and you're going to see cell membrane and be like, we've never even discussed cell membrane. They're the same thing. So I put all the possible names, all the potential ways it could be referenced here. So if you study it like this, you'll read it in a question, and you'll know that it's all the same thing. Now, where will I find these lipid bilayers or cell membranes? On the cell. On every cell. Meaning, if you're alive, you have a lipid bilayer. And you just happen to be composed of over 100 trillion cells. So guess how many lipid bilayers you have? Oh, 100 trillion. Yeah, forever how many cells you have, that's how many lipid bilayers you have. Is this the type of fat that you can work off and burn? No. Okay, and we'll discuss that on Thursday. So this lipid bilayer is composed of individual units. And these individual units are called phospholipids. They're called phospholipids because they're half fat, so that's the lipid part, but then they have a phosphate head, so phospholipid for one unit. The phospho part is polar, which means what? It's hydrophilic. It likes water. And that's good because what do all living organisms have to have? Water. But the lipid part is nonpolar, which means it's hydrophobic, which is crazy because we have water in us, and it would make sense for us not to have something that's hydrophobic. The whole purpose of this structure is to regulate the water contents and what gets in and out of our cells. Okay? That's the purpose of that, and I'll teach you that. But we need part of it to be hydrophilic because we have water all throughout us. So you'll notice that there's hydrophilic here. And it's protecting the hydrophobic and creating that membrane. It's a bilayer because if it was just like this part when exposed to water would do what? It would not mix. It would continue to try to separate. This is going to sound crazy, and some of you are going to get it, some of you aren't, and I respect it. How many of you have seen Big Hero 6, that movie? Okay? If you haven't, it's okay. This is one of those 10th wonders of the world. But this right here, I was like, what do you mean, like it does this? Big Hero 6, they had those little magnets. Okay, those little magnet robots. Those are the equivalent of one phospholipid. When that magnet robot was sitting there uncontrolled, it did absolutely nothing. But when they put that helmet thing on, that controller on someone's brain, and their brain thought something, those magnets immediately did what? Built whatever it was that they were thinking. This phospholipid laying here will do absolutely nothing. You could have a whole stack of phospholipids and drop them here, and they will do nothing. But you take those phospholipids, and you put them into water, and immediately they go, and they line up like this. Because they're trying to protect the hydrophobic. They literally run into this organization to protect the hydrophobic. It's crazy. Watch it on YouTube. It's crazy. I was like, what? It's crazy. But it's awesome. Phospholipid bilayer, cell membrane, plasma membrane, biological membrane, lipid bilayer. It has a hydrophilic aspect, hydrophobic aspect. Ultimately, this structure controls what enters and leaves the cell. It's half hydrophilic, half hydrophobic. Here's, in words, everything that I just said. So I realize I always use pictures to teach, but I like to include the words too in case you don't get everything that I said there. But it mentions that there's one polar and one nonpolar end, and it forms a phospholipid bilayer, where part of it is hydrophilic and it faces the water, part of it is hydrophobic and it is sheltered from the water. Everyone okay with that? Okay, hydration layer, still on water, no surprise. You already know this, you just didn't never call it a hydration layer. Logically speaking, you hear hydration layer. What does it make you think? Okay, hydration, you're drinking water. Hydration layer. A layer of water. A layer of water. Literally, this is all it is. If you've ever made Kool-Aid, lemonade, tea, anything like that, mixed salt and water, you've done this. A hydration layer happens when you add something to water and it dissolves. Okay? Let me explain using this example. Still sodium chloride. And what's sodium chloride again? Salt. We add salt to water. And what does salt do when it gets in the water? It dissolves. The way it happens is slowly the sodium and the chlorine molecules separate and they are surrounded by H2O. What's H2O? Water. So it's a layer of water. It's dissolving that salt by creating a hydration layer around each atom. That's hydration layer is the way we say it's dissolving. When you were taught this, probably, you were taught that water is the universal solvent, which it is. So water is the solvent. Whatever it is that you're mixing, whether it be salt, sugar, Kool-Aid, lemonade, tea, that's your solute. And when you mix a solvent with solute, you create a solution. And that's by means of a hydration layer. So hydration layers occur as those molecules are dissolved. Does that make sense? Still on water, but now getting to pH in the last topic that we have. Water here is H2O, and everybody recognizes this as H2O. But when water is not water, it's actually divided into these two ions. Which one of these is a cation? Which one of these is an anion? And the H plus is called a hydrogen ion. It's a cation. And the OH negatives are referenced as hydroxide ions, and they're anions. When we look at a solution and we say it's acidic, it has a whole bunch of hydrogen ions. And if we look at a solution and say, oh, that's basic or alkaline, that means it has more hydroxide ions. But what happens when I have the same amount of hydrogens and the same amount of hydroxides? What do I have? Water. When I have the same amount of hydrogens and hydroxides, I have water. But if I have slightly more hydrogens, it's acidic. And if I have slightly more hydroxides, it is basic or alkaline, as another term that you can use for that. So this distinguishes between acids and bases. Okay? Which is a property of water, which is why we're covering it here in Chapter 2. Here it specifically addresses acids and bases, telling you that acids release those hydrogen ions, so creating an acidic solution, and bases will release hydroxide ions, creating a basic or alkaline solution. Now, in you, you're constantly trying to maintain that H word. What's that H word? Homeostasis. And your blood pH, which is where we talk about acidity, alkalinity, should be around 7.2 to 7.45, in that range somewhere. So it's slightly basic. Your blood is slightly basic. But if you haven't eaten in a few hours, and let me rephrase that. Some of you might not have eaten breakfast, and you haven't eaten yet, and you're not eating until after class. What happens to the pH of your blood as you go on not eating? Does it become more acidic or more basic? Acidic. So as you go on not eating, it becomes a little bit more and more acidic. If your blood becomes too acidic, what will happen to you? You'll expire. We talked about a feedback mechanism where I tell you I have a problem and you solve it. What type of feedback was that? Negative feedback. You have receptors all throughout your body that are constantly monitoring so that you maintain homeostasis. And it says, hey, Jessica, you haven't eaten breakfast today. You still haven't had your snack. Your blood pH is becoming slightly acidic. No fear. I'm not even going to alert you about this. I'm going to go ahead and start releasing hydroxide ions so that it does what? If my blood's becoming too acidic, it starts to bring it back to homeostasis. So as I went on without eating, it became more and more acidic. and I'm trying to balance it out to maintain homeostasis. So it's releasing these hydroxides. And it's going to continue to do that until it can't anymore. When it can't anymore, then it's going to let me know we have a problem. It's going to give me a headache. It's going to make my stomach. It's going to tell me I need to do what? Eat. And if I continue to ignore those signs, eventually I will expire. Right? Okay? Your body is in constant battle trying to maintain homeostasis. So here is the pH scale straight out of your book. pH, when the number of hydrogens equals the number of hydroxides, is water. That pH is 7. That's neutral. If that number is higher than 7, it is considered basic. So from 7.1 to 14, it's basic. If that number is smaller than 7, it is considered acidic. So 6.9 to 1, or 0, not 1, sorry, 0. And here are some different substances that fall along that pH scale. Again, pure water is where the hydrogen ions are equal to the hydroxide ions. If the pH is less than 7, it's considered acidic. If the pH is greater than 7, it's considered basic. Why do we have to learn all of this chemistry? Because if a cell becomes too acidic, we will need to maintain homeostasis by changing the environment to be a little bit more basic. So we add bases. If it's too basic, we add hydrogens or acids. We call this buffering, which is why I titled it buffering. And why did I reference a cell? If a cell. What is a cell? It's the basic unit of life. So here, everything that I've talked about, pH, water, and all of that, all applies to cells, and cells are the of life. And our goal is to help keep cells close to a neutral pH. And again, cells are the basic unit of life. So if I can take something like chemistry and tie it to biology, it'll help you learn this content. Or at least that's my goal. For some of you, you're like, no, it's not. And that's okay. Some of you, it's working. This is the end of chapter 2. Does anyone have any questions or need clarification? All right.